Like Dr. Akash said, one size does not fit all. He ended his talk there. I would take it from there, and I would like to now address. We are talking about precision, personalized nutrition, and we are in a different era of nutrition today. And there's a lot to be spoken, more than macros and micros today, I personally feel. And as we were exploring into the era of genomics, post-human genomic study, we have now concluded or understood that yes, we need to work on more on personalized nutrition. And today I'm here to talk or sensitize, I should say. 20 minutes is not enough to in detail to talk about this, but I would like to sensitize the audience here to understand the difference between personalized precision, gut flora, and its impact on vascular health is the topic today. But we all know that somewhere it is definitely end of the time we could link it to kidney disease at the same time. So to begin with, what is personalized nutrition? Absolutely a tailor-made nutrition plan. But if you look at it in detail, it has exosomes, that is the environment. It is your phenotype, and now the genotype. So we have three areas to look into personalized nutrition. And if we are looking at personalized, if we have three reports of that person with us, I think the nutrition becomes more tailor-made and more precise to be of the fact. So precise nutrition is again going more in depth of personalized and looking at specific to food or to nutrients to that area of health. For example, today we are talking about the role of methylation, homocysteine. That's one of the examples which I would be highlighting today. So that is what is precision nutrition. We would be look at it. So nutrigenomics and genetics, these are the two areas which have been explored because it is linked with genomics and nutrition together. We call it as nutrigenetics, nutrigenomics, and we call a counselor who talks about this as nutrigenomic counselor. But a molecular biologist or a molecular science has a more important role to play, and nutrition is an application part of it. So genetics and genomics is just two sides of the coin, as already said. Genomics is response of individual genes to nutrients and its metabolic consequences. And genetics is a personalized nutrition or a different phenotype to a specific diet. And exposome, as I've already said, is an impact of environmental. I'm sure all of you agree today, I'll be talking about all this in detail for next 15 minutes, that we all are made up with genetic constitution, but the challenge is epigenetics. What is playing more important role on our genes is the environment, or we can call it exposome. We do know about anthropometry, biochemical, clinical, dietary, and if we add a factor of exposome and genotype to it, I think that contributes to a better nutrition prescription because you are looking at an integrated approach towards your personalized nutrition prescription. So today, I feel that we are now into an integrated systems biology, from soil nutrition to the cellular nutrition, where you have the plant science, the genomics, the genetics, the microbiology, everything having an interplay, and we have bioactive components of the food and the lifestyle modification environmental factors to help us learn ourselves better. So it's not as complex as I'm making it view to you. It is very simple way of looking at it. A nutrition prescription today, I think, is going way ahead than saying 60% carbohydrates, 20% protein, 20% fat. It is getting into details by showing that which carbohydrates do I need? Is my body tailor-made to work better on an insulin resistance diet with a resistant starch, with some kind of a prebiotics in my diet, or I'm more easily prone to adipogenesis with a higher carb load in my body? I need to know that 2 chapati sabji or 1 chapati, 1 sabji, 1 dal, a glycemic load of the calories are same, 1600 calories all of us prescribe. Going more deeper into the calories is the composition of my calories, of macros, where I should know that I need a glycemic load, for example, or it's just 2 chapati and bhaji which contribute the same calories. So that's what I'm trying to understand, that how does my body drive to adipogenesis, to insulin resistance, to fat deposition, to oxidative stress, and n number of factors. So I think this is what we are trying to learn with the help of this personalized health, that the influence of nutrition on a particular gene. So we are now in an era of omics, friends, where we have the genomics, proteomics, metabolomics transcripting us into a newer area of disease prevention and preventive health. And I believe this is all about learning molecular nutrition and a synergy pathway between the food, the nutrients, the gut flora, and the exposome, which is environment. How wonderfully we train our body to handle exposomes. It may be stress, it may be food, it may be circadian rhythm, it can be n number of things. And I think today the dietitian should be well equipped with this knowledge of the post-genomic era tool, which we have to tell us about the genetic background. We need to understand the knowledge about omics technology, where at a protein level, at a DNA level, when my cell has an interaction with a nutrient, for example, I would say that a B12 or a folate, then what my body's behavior towards that particular nutrient is. Do I have a strategy in diet which can target to optimize the nutrition? As one size does not fit all, I today would like to maximize my nutrition. I want to get the best out of food which I'm eating for my own health. My RDAs, my EARs tell me how much to eat, what is required. But my body decides how much to absorb and metabolize. And if I learn this nutrigenomics or nutrigenetics application of nutrient-based nutrition, and I think it will help us to associate our health with diet, with prevention, with wellness, and with diseases. And there is a recent article on the paper in Journal of Nutrition 2020, where it has been highlighted that precision and personalized diet plan for kidney health and disease management. When we talk about so much of macros and micros, and I've been hearing this since 2 days, are we trying to restrict food so that it is not helping my gut flora to have a better metabolism? We need to examine the potent role of diet in modulating gut microbiota composition. While I'm restricting vegetables and fruits, am I restricting a lot of prebiotics in my diet for my gut flora to enhance a good flora to work better on nutrient modulation? I want this august audience of nephrologists, physicians, dietitians to understand and to work on saying that how I can give the balance and optimum nutrition, which will also work better on my gut flora. Because plant-based foods have a very important impact on improving my gut flora. I have my own probiotic, which is being synthesized from prebiotic. So the prebiotic needs to be tailor-made. I'm sure we are not far away from the era where now with this tool examination, we'll be able to identify what prebiotic my body can take in so that it gives a good flora to what I would like to highlight for the better absorption and metabolism. So KDOQI guidelines implemented in accordance with principles of precision nutrition, including a personalized approach to intake of the dietary protein and other macro and micronutrients. So I'm sure you would all now start thinking that we have our guidelines of macros and micros in place in CKD, but can we start looking differently to work better so that we're not restricting or we are not suppressing a good gut flora in our system? Let's take, for example, a case where we have a 35-year-old middle-aged, to talk about precision nutrition, we're just highlighting a case, who is detected with essential hypertension. And we know that nowadays the challenges with lifestyle, which contributes to essential hypertension, are all this. A typical nutrition prescription or a lifestyle management prescription will be fat loss, behavior modification, lifestyle modification, work on your diet, prevent it, And even the physicians say, go back, first work on your diet, lose weight, and we'll monitor your hypertension, and we'll start the medications. Now, if I have this precision nutrition tool in my hand, how will I do it differently? I can think more detail about this patient into a different way of looking at his micronutrient management, at his vascular management through hyperhomocysteemia. Yes, weight loss is my primary, behavior modification, fat loss, all that will come parallelly, but some different angle to my prescription, if I can look at it, is can I look into homocysteine? Because we all know that hyperhomocysteemia diminishes the vasodilation by nitric oxide, which increase oxidative stress. Let us take these two words, nitric oxide. We know that's the precursor of arginine, and hyperhomocysteemia, and how does my prescription differ in a personalized nutrition? And let's little dwell under methylation chapter, our folic acid cycle. And we all know that we have methyl tetrahydrofolate reductase, the MTHFR, which helps me to convert folic acid to folate. If I do not have, I take in dietary folate, fantastic. Otherwise, if I am having a mutated or a variant MTHFR, my body refuses to convert folic to folate. And we all know that if I have a poor folic acid conversion, and my folic acid conversion needs B12, B6, pyridoxine, B2, right, to get into a good folic acid generation cycle of homocysteine. And this, I'm not going to details of cycle, but I'm trying to highlight that folic acid, one folate molecule, if I do not get, and along with it, if I do not complete the cycle of folic acid, I do have a hyperhomocysteemia tendency, because I also need a B6 to convert the homocysteine into cystathione and a B12 molecule to get it back to methionine. And if I know my genetic mutations or variations of MTHFR, and then my prescription will change to folic acid rich foods, supplements, folates, B12, B2, B9, the betaine molecule, which is also important for your neurotransmitters like serotonin. And my methylation map goes in 4 cycles. I have a urea cycle where we all know that in urea cycle, the nitric oxide, which we all look at it as oxidative stress, are a potent vascular nutrition precursor arginine. First is urea cycle. Second is my neurotransmitter BH4 cycle, my folic acid cycle, and my methionine cycle. And from this cycle, I know that how important my B complex vitamins are and how important is my folate and B12 is to work better on my vascular health. If my prescription of nutrition goes with folate rich, lysine rich, methionine rich, lysine are legumes, methionine is rice, along with the present prescription of lifestyle modification of whatever we say. So I think the way we start looking into a personalized approach changes to working from nutrient-based nutrition like Nutrigenetics application of active folate, active B12, B6 supplements, magnesium, trimethylglycine, one of the amino acids, vitamin D, to work better on methylation cycle, because homocysteine is also a clinical area where we can first check that if the patient has hyperhomocysteemia, we look at the genetic report of methylation. Can we start looking at this holistic approach and work better on nutrient-based nutrition? Because the last point which you see here is genetic mutations in your methylation enzymes, MTHFR, CBS, COMT, that is MTR, all this will definitely help. So enhancing a good gut flora to have a better absorption of vitamin B complex and complete the various metabolic pathways in our body is absolutely the need of nutrition prescription today, along with micros and macros. There are foods to support B6, B12, the glycine amino acid, the methionine, and the amino acid, which is complete in animal proteins. And exercise also through circadian rhythms can be worked on your DNA or methylation cycle. Because if you are having mutated MTHFR, aerobic or resistance training nearly 30 minutes per day for 5 days a week recommended. Of course, this goes very personalized. We're just looking at a hypertension management and an MTHFR. If the patient goes into a CKD, a different way can be looked at it. So circadian rhythm, your body clock, time of exercise, the way your neurotransmitters behave with you, all this can be thought of in a precision or a personalized nutrition, because you will have bioactive components of food. For example, you have glycine, you have choline here, and you have B12, you have folate. All this in those 4 methylation cycles which we have seen will help us. This is how the genotype report will look like, friends, where you have a methylation cycle of certain genes. These are all studied through human genome cycle. They come into preventive genetics, not the predictive genetics, where the role of nutrient-based nutrition is being highlighted here. You have oxidative stress, where you have INOS, SODs, which will help us to learn what kind of precursor do you need. Do you need arginine? Because we all know arginine is a precursor for INOS, and we do many times give, and my nutrition supplement or support also can be worked accordingly. Lifestyle management for this patient, where I can highlight some important genetics like PPR gamma, FTO, TCLF, and these are all research genes. And if I know that they have variations in it, my prescription changes, like I said, in between the glycemic load concept of the meal, the macros distribution in my food, the prebiotic composition of my food, all this changes because I know the role of each genotype, which actually tries to work better on the expression of the genes. So if I play with epigenetics, I can be able to modulate my gene expression. And I think that's another way of looking at your nutrition prescription, with few factors to be highlighted. Because if you have this kind of a patient, which I've just shown you in the beginning of the slide, you can't deny the role of exposomes, which is more higher, the lifestyle modification. The exposomes contribute to inflammation in the body, and maybe the fat also is contributing to inflammation. Giving a high protein diet, a keto diet may not work. We're talking off CKD here. May not work. Maybe if the inflammatory genetics demand an anti-inflammatory diet, like IL-6, TNF, then I need to go with a simple anti-inflammatory low-calorie diet for that person to make him lose weight. So which diet prescription, when diet prescription, how much to eat, I think all this is being reflected in this genotype information. I definitely say that this is 10 to 20% of your total contribution to your prescription. 80% is your, again, your own lifestyle management. But one aspect added to this we cannot now forget is A, B, C, D we are already doing. E is environment, G is genotype. I think these 2 areas we have added more into detailing of a diet recall of the patient. And maybe there are its own challenges. It's a little expensive to get into genotype. Finally, what will work is motivation. Finally, what will work is compliance of the patient. And that is why I think through this, the only way of me getting into this forum and sensitizing the audience is when we describe any diet plans with a stricter or restricted modifications of macros and micros and hydration, are we also not looking at the gut flora? Can we start more detailing into good amount of plant-based foods for ample prebiotic support to the patient? And finally, ethics, training, and integrated approach is a way forward. I thank chairpersons and the audience to be a patient with my session today. Thank you so much.